Okay, so I am going to be talking about our combinatorial work, and I will preface this by saying that we don't necessarily study the antidepressant effects of ketamine. We actually study the prophylactic effects. And so these are our disclosures. Just one of the ones that are applicable today is a sponsored research agreement from Silo Pharma. So I think you've heard from Adriana and everyone today that in most cases, patients are stressed. They then develop PTSD or depression, and then we're giving ketamine or drugs after to deal with the symptoms. Our lab had a serendipitous finding. We did not set out to do this, where we found that giving ketamine 1 week before stress actually totally prevented the effects of that stress. And so our working model is that you can make small molecule compounds that you give before stress in cases where you know stress will occur, like military personnel, that allows for long-lasting protection after or if a stressor were to occur. Now, I know this is not realistic. We are probably all stressed at different points in our lifetime. If we're in a car accident, we don't know that we're going to be in that car accident. And so we're trying to use this as a model to better understand stress resilience versus susceptibility, but also to make drugs that you could give right after the stressor and not wait for the patient to fully develop PTSD or depression. And so obviously, we're all talking about ketamine. I'm interested in the actions of ketamine, but I'm also interested in using ketamine as a pharmacological way to better understand what are the circuits mediating resilience versus susceptibility, but in a controlled setting where you can make most of the mice resilient with ketamine or most of the mice susceptible with stress. So the first study, now published many years ago, was done by two graduate students in my lab, and Adriana knows the story probably all too well. We were using ketamine as an anesthetic when I was a grad student and looking at neurogenesis, and we kept having problems with the control mice. Fast forward many years, we end up developing this paradigm where I'm going to try to get the laser pointer. Okay. Mice are injected with a single injection of saline or ketamine, and then 1 week they're administered a stress. And here, oh, I'm going to switch. Oh, perfect.
Just press that top button and
Okay.
hold it
Okay.
while you're pointing.
Okay. Let's see. Can everyone see that? Okay. There we go. Okay. So they were administered a social defeat stress. This stress is a common rodent model where animals are put in an environment where they get beat up each day, and then you separate them. And then we administered a number of assays after the stressor. And what we found is that in the animals that got control, no stress, there was no difference if they got this single injection of prophylactic ketamine. But the animals that underwent the social defeat stress had increased immobility or increased behavioral despair in the forced swim test. And the animals that got this prophylactic ketamine injection now 22 days earlier were protected against that stress and back down to control levels. We were able to replicate this or extend these findings to a social assay where we looked at animals after social defeat, and they usually hide from the aggressor. And here what you can see is that the animals that got prophylactic ketamine and were socially defeated, they spent less time hiding out behind this empty cup. And you can see this in this pink or purple lines where the social defeat mice are hiding out behind the cup, and the animals that got this prophylactic ketamine are walking around the arena. So it works against the behavioral despair, and it works as well for social avoidance. We didn't just do this in social defeat. We verified it using other stress models, and one of the stress models that we used was learned helplessness, in which animals get 70 shocks in a box, and they develop helpless behavior. And we can find with ketamine, again, single injection 1 week before. I'm sorry, there's like a delay in this, and so I'm screening, and it's like over here. I'm not going to point. The animals that got prophylactic ketamine, they were basically less helpless or had a decreased latency when compared to the control animals in the learned helplessness paradigm. And when we used a contextual fear conditioning assay, animals also had a blunted fear response but did not have differences in extinction if they got a prophylactic ketamine injection. So here are a number of different stressors I'm telling you about, but I do want to point out is that my lab does not use the canonical C57 mouse strain that most basic labs use. We use a 129 line, which is obviously very susceptible. They are not aggressive. They're kind and sweet and a little anxious, but we want to have a level in which the animals are low and susceptible so that the drugs can bring them up. We've verified this also in C57 mice, but I think it's an important point to make when we talk about using rodent studies. If we're looking at stress and susceptibility, we should not be using the most resilient mouse out there, the C57 probably. So we had another postdoc in the lab who wanted to verify these findings and extend them, and we made use of a paradigm called a contextual fear discrimination paradigm. Adriana can probably also tell you about this better than I can. In rodents, we try to model fear overgeneralization with a contextual fear conditioning task and different contextual cues. And so what we do is we give animals a brief shock in a context A, and they get a brief shock every day in context A. But they also get exposure to context B, which looks like context A, but is slightly different. And over time, animals will decrease their fear to the context B, the neutral context, and increase or keep their fear constant in context A. And this task is supposed to model fear overgeneralization that you see in patients with PTSD, even late life depression, where animals don't basically compartmentalize their stress into one context. It gets generalized between the contexts. And so this postdoc, Alessia, wanted to understand, could prophylactic ketamine also protect against fear overgeneralization? And so the animals got one injection of saline or ketamine a week before this contextual fear discrimination task, and then they got shocked for 10 days in A, or in context B with no shock. And what you can find is that the animals that got prophylactic ketamine, they actually learn the task much more quickly than the control animals. They decrease their fear and do not generalize it. By the end of the task, both saline and ketamine can actually separate their fear, but the ketamine animals learn it faster. Now, we didn't publish this in the paper. We've been holding on to it, but we had this finding where we looked 6 weeks after the initial ketamine injection. One injection, sub-anesthetic, 6 weeks later. And the animals that got prophylactic ketamine still do not generalize their fear at this time point. You can see here in the dark red versus the bright red, whereas the control animals do. And that's what you would typically think of seeing at a remote memory recall test, where animals generalize. For whatever reason, 6 weeks later, a single injection of ketamine allows these animals not to generalize their fear. So this is the part I put in for Sarah, and so I'm glad I have a couple extra minutes. We recently published a paper last year. It came online in January in Biological Psychiatry, and we wanted to see if we could apply ketamine in a mouse model of TBI. This is a joint project we did with Steve Kearney, who's also at Columbia, and Josie, who was my grad student and now is in Bill Carlazon's lab at McLean. So there are a couple moving parts to this. We wanted to model TBI in mice. We wanted to see if they had a fear overgeneralization phenotype like I just showed you, and we wanted to see if ketamine could help it. The other moving part is that we made use of a genetic line to tag individual memories for the life of the animal in the animal. And these are mice that I engineered. It's the other part of my lab, the other part of my life. They're called the ARC-CRT2 mice, and they allow you to tag one memory with single-cell resolution across the brain in the life of the mouse. You can then look at this single-cell resolution of a memory in any disease state you want, but here we applied it to use it with TBI. So animals were given a TBI procedure. Here we used a cortical controlled impact. They were then left in their home cage for a month, or they had a sham procedure. We had administered a number of behavioral assays to look at what TBI did, and then we administered this fear overgeneralization phenotype or behavioral paradigm that I just mentioned. The important part here is that we tagged the neural ensembles that were active during the encoding of the fear, and we also tagged or visualized the ensembles that were active when they expressed the fear. So we have 2 tags in the same mouse to look at how does TBI affect how these animals are encoding the fear, and how does TBI change or alter how the animals are expressing their fear. So what you can see, it's easier if you look at the bar graph. The animals with TBI, they're terrible at this task. They overgeneralize their fear. They don't separate the freezing in context A, the aversive, or the context B, whereas the sham animals can do it actually pretty quickly by day 6, and they have more freezing in context A, less in B, and the TBI animals are equal. So we went in and we looked at these individual memories of the fear encoding, of the fear retrieval in these mice. And the tag for the encoding is EYFP. The marker for the expression is CFOS. And the colocalization of the cells that are active at both time points are what we call a memory trace or an engram for this fear experience. And what you can see if you focus down here is that animals that got sham, no TBI, and were re-exposed to context A, they have high-low overlap of these ensembles in context A, but low in context B. And the animals that got TBI, there is no difference between these ensembles. This is in the dentate gyrus. We did it in the amygdala, in the PFC. And what we find is that animals that have fear over generalization, they have improper reactivation of the fear memory in CA3, in the dentate, in the amygdala, and this could be what's driving this improper behavior in the animals with TBI. Now, I'm not going to go into that much with this, but we wanted, and why I'm showing it today, is to test what ketamine could do to this phenotype. So we have a mouse model TBI, they overgeneralize their fear, we know the memory traces are altered in the dentate gyrus, what can ketamine do? And so what we did is we gave animals a CCI procedure and 1 hour later an injection of saline or ketamine. We then did a bunch of behavioral tasks, including this fear overgeneralization task. And what we found, if you look at the bar graph on the right here, is that TBI mice that got saline, no ketamine, they overgeneralized their fear. But animals that got the single injection of ketamine right after the TBI procedure, now they can separate where they freeze more in A and less in B and don't overgeneralize their fear. So we looked at the memory traces again in the dentate, and what we find is that if you remember in the last graph, the TBI animals, they don't separate freezing in A, freezing in B. They have the same overlap between the memory traces. Now the animals that got ketamine, there's a separation. They have a proper reactivation of the fear memory and not an improper reactivation of the neutral or negative memory. And so we have behavior that lines up with these ensembles in the dentate gyrus, and ketamine can actually fix it. So that is just, I try to give you a little primer on prophylactic work in the lab. We obviously are not doing antidepressant, but we believe that this could be a novel technique to protect at-risk patients. And if we can understand these mechanisms, could we develop other things? And so I just want to summarize what I've tried to fit in 10 slides right there. Prophylactic ketamine, single injection, it works in 6 different stress models that we've looked at. It's really effective at behavioral despair, at social avoidance, at learned helplessness. It attenuates fear but does not accelerate fear extinction. It decreases fear overgeneralization. It's really bad at anxiety-like behavior in rodents. It's terrible at context fear extinction. We don't see changes in locomotion long-term, and we don't see changes on hyponeophagia. The prophylactic dosing is different than the canonical antidepressant dosing in rodents, and it's also sex-specific. We get this prophylactic effect to work at 10 mg per kg in females and 30 in males. We've looked across the lifespan. I've shown you adult mice here. We can get it to work in adolescents. We cannot get it to work in aged mice. We've published 2 papers now. It lines up with the Janssen-Spravato early results. We cannot get it to work at all in aged mice. So what I'm not going to have time to tell you, but I'm going to tell you about hopefully 1 or 2 other little projects, is, I love talking about ketamine. I'm really happy to be here, but I don't think ketamine is, the holy grail for prophylactic work. And so we always wanted to find a suite of compounds that could let us understand this mechanism. Like, how is it that you give a single injection of a drug, you protect against stress, and you have these long-lasting effects. And so we've expanded now, and we have the metabolites of ketamine. This was published also. The 2R6R HNK works really well for behavioral despair, but the 2S6S HNK works really well against fear. And so when you put them together, we think this is how we're targeting fear and depression in the rodents. We have a new molecule out that's been published called FENM, 3 5-HT4 agonists, which I'll get to today. We have 8 new compounds that we've been in development with Don Landry and Jonathan Javich at Columbia. And what I'm going to hopefully end with is this combinatorial treatment of combining some of these drugs to target phenotypes that could not be targeted with either drug alone. So I gave you a lot of mouse stuff today, and I apologize because there's a lot of clinicians in the audience. But believe it or not, we published this. We thought no one's going to pay attention to this. And then all of these studies have come out trying to extend our findings into humans. And Sarah is one of them with many papers. So there was a study which I think is one of the more interesting studies that I was not aware of when we first did this study. There were Iraqi war veterans who were burned on the battlefield, and they were given ketamine right after the burn. And what they found is in the war veterans that got ketamine on the battlefield, the prevalence of PTSD dropped 27% from the no ketamine. This wasn't replicated in a follow-up study, but since then, a number of other groups have tried to extend prophylactic ketamine to postpartum depression, S-ketamine, and now Sarah's doing it in healthy volunteers with James Murrow and Dana Charney at Mount Sinai. So it's not just in the mice. So what is the mechanism? Like, how could this possibly work? How has no one figured this out, that a ketamine could have effects that last for at least 6 weeks, if not longer? We have not looked past there. And so one of my postdocs, Alessia at the time, with A.J. Robinson, who also used to be at Mount Sinai, decided to try to target different ensembles to figure out what could be driving these effects. And so Alessia bilaterally injected a virus into ventral CA3. Ventral CA3 is a subfield of the hippocampus. We had a lot of data suggesting that CA3 was a prime target in prophylactic efficacy. And then she injected saline or ketamine and administered a fear test and a number of other behavioral assays. And when Alessia blocks the transcriptional factor delta phos-B in ventral CA3, I want you to look at the bar graph on the right here. In control animals, we get this attenuation of fear that is consistent with prophylactic ketamine. But in the animals that got this virus to block delta phos-B in ventral CA3, you can completely block the effects of ketamine by just targeting ventral CA3. Now, what I'm not showing in this graph is that she also did the opposite experiment where she overexpressed delta phos-B in ventral CA3. She could completely mimic the effects of ketamine by just targeting ventral CA3 activity. Now, this is in line with a lot of Todd Gould stuff, looking at CA1 in the hippocampus, thinking it's presynaptic. Well, CA3 is presynaptic to CA1. And so we really believe that CA3 is a key node in the efficacy of prophylactic ketamine. Since then, Alessia has tried to expand, just looking at the hippocampus, and we've developed a brain-wide pipeline to visualize these memory traces or whatever experience that you want with single-cell resolution across the brain. This was done by Sophia and Marcos, 2 MD PhD students, and Sophia's back at Columbia now in Renee Hen's lab. And so they develop code and alignment to clear mouse brain tissue to make it see-through, and then you can basically image the entire brain with single-cell resolution. And Sophia first, as a proof of principle, used propranolol to do this, but you can basically get, a behavioral assay, a behavioral output, and then with single-cell resolution, build these neural networks that allows you to identify different nodes that may be altered with the drug or whatever treatment that you're given, and then look at how these treatments affect the neural activity linking together. Alessia is currently doing that right now with ketamine and one of the metabolites of ketamine. And so I mentioned earlier that this 2S6S HNK robustly reduces fear, but not depression in rodents. And so she's doing a comparison, it's in revision, of saline versus ketamine versus the 2S6S HNK for brain-wide single-cell resolution of how these ensembles are changed. And you can see that although the behavioral output is the same, where ketamine and HNK both decrease fear, their neural networks are not, and we see divergence in the neural networks across the brain. So the last piece of data that I want to give into the mechanism is we obviously know that CA3 is important, but it still doesn't justify how this drug is having such long-lasting effects. And we did a study where we injected saline, ketamine, or one of the HNKs, and then a week later did slice electrophysiology in ventral CA3. We looked at, passive electrophysiological properties of the cells in CA3 with Victor Luna, who's now at Temple, and Brianna Chen in my lab. And what you can see in the saline animals, we have these large AMPA bursts in CA3. Ketamine, they're totally gone. The metabolite that works for the depression, the 2R6R HNK, they're totally gone. The 2S6S HNK, they're back, and this is the metabolite that doesn't work on depression. Every single other drug we have tested as a prophylactic, if it works, these AMPA bursts are gone. If it doesn't work, the AMPA bursts are still there. And so we think this is a key signature of whether you want to call it resilience, pharmacological resilience, and if these drugs are working. And so now we're able to screen them relatively quickly. The 5-HT4 agonist, the novel molecule, some stuff I'll show you later, all of them get rid of these AMPA bursts if they work, and if they don't work, the AMPA bursts are still there. So I'm going to try to, how am I doing on time? Okay. So I'm going to show you some of the other stuff outside of ketamine. I still love ketamine, but we have a novel NMDA receptor antagonist that is basically a derivative of memantine, and we are working with Gilles Rubenstein at Rest and Brianna Chen, my postdoc, and this came out now 3 years ago. And what we have tested is, can FENM also be a prophylactic or an antidepressant? And what we found is that the 20 and 30 mg per kg single injection of FENM 1 week before stress attenuated fear, decreased behavioral despair in the forced swim test, like ketamine, but not like memantine, its parent compound. And I'm not going to show it, but what's great about FENM is you can give it also right after the stressor. It doesn't have to be a week before, and so that opens up that window of, if you don't know a stress is going to occur, but you want to protect against it, could you give it right after? The other things that we've been working on, and again, this is also I don't want to say serendipitous, but it is a serendipitous finding of having really good friends. We had tried for years to try to get fluoxetine to work as a prophylactic, and I kind of just discounted the whole serotonergic system, like, oh, it's not anything to do with serotonin. And our really good friends work on 5-HT4 and begged us for years to try some 5-HT4s as prophylactics. And I kind of hit the wall of I'm done testing drugs, we're just going to go with ketamine. But they were right. And I'm showing 2 up here, but there are 3 that we've tested initially. One of them is Prucalopride. In the US, it's FDA approved for chronic constipation. It's a 5-HT4 receptor agonist. And we found that in a single injection of Prucalopride, this other drug, which is also a 5-HT4 agonist, PF, I'm not going to list all the numbers, and the more commonly used 5-HT4 agonist is RS67333, all are effective as single injection prophylactics, much to our surprise. They decrease fear and they decrease behavioral despair pretty robustly in the mice. So I'm going to stop here for a second. This is great, right? In mice, we could protect against stress. How is that going to work? But if you've paid attention, I've only really talked about depression and fear behavior, and I really haven't talked about anything else. And so we set out to try to figure out, could we target some of these other endophenotypes that we could measure in mice that aren't really working with these prophylactic compounds. It doesn't really change anxiety behavior. It doesn't change feeding behavior. How can we start to target these other symptoms and then make it applicable to other patient populations? And so we had the idea to combine ketamine and the 5-HT4 agonists together and see if this would potentially work. 5-HT4 agonists have been known to involve anxiety and feeding behavior, and so we thought maybe together the surge could help. These graphs had a lot of bar lines, and I put them on there for completeness, but we injected saline, ketamine, Prucalopride, or a combination, and then mixed and matched the doses 1 week before stress. And a single injection of each works against fear, and a dose together works against fear. Single and combined dose works against depression. That's not really something great, but when you look at this test called the novelty suppressed feeding assay, which looks at an animal's drive to eat in a really anxiety-provoking environment, we call it hyponeophagia. Now, out of nowhere, we've never had an effect on this test before, ketamine and Prucalopride decrease the latency for the animal to eat, and they eat much more robustly in this paradigm. And so this is the first time in all of these drugs we tested do we start to get an effect on anxiety-like behavior or feeding behavior only when they're combined together. This is males. We've done it in females as well. Again, single injection or combination injection 1 week before stress. We can really affect depression. None of the drugs I've talked about today really affect fear in the females. But when you look at the hyponeophagia behavior, now a single combination dose of the two drugs again decreases the latency for the animal to eat in the arena compared to control animals. So let's go back to the AMPA bursts. And going back to the AMPA bursts, injection of drug, 1 week, brain slices, slice electrophysiology. AMPA bursts are there in the control. AMPA bursts are gone in the combination. So I hope this gives us some hope of it's not just all depression and fear, but we are really, I'm going to fast forward a minute so that I'm not going to run out of time. I'm hitting the wrong buttons, I'm sorry. I'm going to skip past this. But, this is all, again, prophylactic. How realistic is that in the clinical population? And so the last thing that we did now is given it after stress. So single ketamine, Prucalopride, K plus P. After the stressor in male mice, we can affect freezing if it's combined. We can affect behavioral despair if it's combined. And that hyponeophagia, we can also affect it if it's combined. And the study I mentioned earlier with Silo Pharma is we're doing it now chronically to try to also mimic what's more done in the clinical population. These are a lot of bars, and I'm so sorry, but these groups either got ketamine once a week, twice a week, different doses, Prucalopride constantly at 3 different doses, and then we mixed and matched the ketamine and Prucalopride chronic or twice a week or once a week. And what you see is that chronically giving ketamine and Prucalopride after stress decreases freezing in the fear conditioning assay, decreases behavioral despair, and what I didn't have room is it also affects feeding behavior. So what I hope I showed you today is that ketamine doesn't just have to also be a rapid-acting antidepressant. It can be a prophylactic. It's really effective at altering fear generalization, fear expression, stress-induced behavioral despair, social avoidance. It's terrible at anxiety, fear extinction, and hyponeophagia when given alone. I gave you a lot of background on the other compounds that we've been working on, the metabolites of ketamine, the novel drug FENM, some 5-HT4 agonists. We have a bunch of other compounds in the pipeline that are working. And we also now have them working together. So hopefully with the phenotypes that we can't target with single drug administration alone, maybe we can start targeting it with them together. And last but not least, by hopefully using, I know these are mice, these are not patients, but they're really useful for getting at the circuits and identifying the nodes. And so I hope by using these cool genetic tools that we have in rodents, we could better understand stress resilience. We can better identify the brain regions that are being targeted to improve stress-induced psychiatric disease, and then make drugs that are more targeted versus one or the other. And I hope I didn't go too much over, and thank you so much, and I'm sorry for all the issues with the PowerPoint, but thank you.
Thank you very much indeed. I'm afraid we're not going to have time for questions.